in Davydov reading, and the next speaker, Sergiy Perepelytsya, DNA, polyamide, interaction, conformation, insight. Please, 25 minutes.
25 minutes. Thank you very much, dear colleagues. First of all, I would like to thank the organizers, and for me, it's a big pleasure to give a lecture, to give a talk about my research. And today I will tell you about DNA-polyamine interactions, conformational insights. So in my presentation, I will tell you some important features of DNA double helix and about polyamines, their role in living cell, and their features of interaction with DNA. And also, what is very important, the conformational flexibility of polyamines. As an example, we use particular polyamine molecules. This is spermidine. And we will discuss anomalous band conformations of this molecule. I just remind the structure of the double helix, the structure of DNA. It is well known that it has the double helix form with sugar phosphate backbone outside and nucleotide pairs inside the double helix. Each phosphate group of DNA is negatively charged, and this is very important for DNA. It gives special features that are very important for biological functioning of DNA. And this structure was discovered by James Watson, Francis Crick, and they used experimental results by Maurice Wilkins and Rosalinda Franklin. And she was the first who pointed out that DNA can have different conformations that depend on environment, depend on hydration of DNA, depend on salt. And here you can see these images. This is image of bDNA. You can see this structure of bDNA that is built according to the modern data. This is a characteristic cross here, and also A structure, which is known now. We know it as A conformation, and the X-ray image is completely different. Why I'm talking about this in so details? It is very important because DNA is flexible and it can take different conformations, and this is the response on environment, on hydration, and also ions. And if we read the paper, which is 70 years old, by Rosalinda Franklin, she put in her paper this scheme where she indicated that the phosphate group should be neutralized by metal ions, in particular sodium ions, that are always present in the solution. This property of DNA is studied extensively. And as I told 70 years ago, you can see these papers. It was in 1953. DNA double helix was discovered, and University of Cavendish published such a picture describing this, dedicated to this discovery. This is Watson, Crick, and also they indicated Rosalinda Franklin. And now it is recognized her contribution to this discovery. The double helix should be stabilized by metal counter ions, but not only metals. It can be also molecular ions, polycationic ions like natural polyamines. This is chained small molecules with amino groups which are positively charged in a water solution. And you can find a lot of different polyamines, but here putrescine, spermidine, and spermine, this is natural polyamines which are always present in a living cell. And it is believed that polyamines play an essential role in many mechanisms of related to DNA-protein interaction, folding of RNA. Polyamines are also present in viruses, and there are a lot of different properties, for instance, like radio protection and so on. The most important property of polyamines is related to interaction with DNA, because DNA, as you know, is folded around protein core in our in the living cell, and it is partially neutralized. The charge of DNA is partially neutralized by protein, but it is not enough to make this system completely stable, and the rest of the charge is neutralized by counter ions. It may be metal ions and also polyamines. And after that, this chain of nucleosomes, this particle is called nucleosome, form chromatin, and after that, chromosome. And that's why polyamines, interaction of polyamines with DNA were studied from very beginning. This is probably the first work, calculation work related to the study of polyamines with DNA, with B conformation, A conformation, and a very long time it was considered that polyamines bind to DNA not specifically. There are negatively charged phosphate groups, and that's why polyamines bind to the backbone, and the sequence of nucleotides doesn't play any role. But the experimental data, not many years ago, show that polyamines bind specifically to some nucleotide sequences, and this problem should be studied. There are a lot of different studies about this. So this is some of them by different groups. And what is important to notice that usually the system of DNA is studied, if we study the interaction with polyamines, it is studied with high concentration of polyamines. So the polyamines are everywhere, and they bind to different regions of the double helix, and that's why it's very difficult to select some specific region where polyamine can bind. In contrast to the previous works, we took the system with low concentration of polyamines. So this is the system with putrescine, spermidine, and spermine, and only several molecules of these ligands were added to DNA. And we simulated the system, and we found that polyamine bind in some specific way. So it goes inside to the double helix, and it finds the narrowest place, the place with the narrowest minor groove, where it's stuck for a very long time. This is the time about 100 of nanoseconds. And what is very important here is that polyamine is almost stretched. So the conformation of polyamine in this case is linear. But there are many other simulations, and in particular also our simulations that we did for other systems. This is the system with 2 DNA molecules, and also the system of nucleosomal DNA, where polyamine bind to DNA, and they make these two molecules together, so some aggregation is observed. And in these places, polyamines can change the shape, and the conformation of polyamines is really important in this case. So we just schematize this interaction in such figure. You can see here the polyamines as some cylinders which are rigid and so on, but in fact these molecules can bend, and this is very important. So what is the role of polyamine conformation? So we understand now, just analyzing the results that we obtained previously, we can say that in some cases polyamines are stretched. They bind to DNA, they are localized in the minor groove, for instance, and they are completely stretched. But in some cases where polyamines are responsible for the aggregation of DNA, they can bend. And in this case, conformation play essential role. And we saw this. There are some results that we publish in supplemental materials to all these papers that I cite, showing that the distribution of end-to-end distance is not uniform, and it means that there are some conformations of polyamines which are preferable in the case of interaction with DNA. So to analyze different conformation of polyamines, we run the simulations of DNA with high concentration of polyamines. You can see here this video describing how the polyamines move around DNA. Actually, they stuck to DNA, and this is very short part of this trajectory, only 4 nanoseconds, about 4 nanoseconds. So they are almost in the same place in some particular region. And to compare these results with solution, we also simulated the system without DNA, just solution with polyamines. And we analyzed the conformation in both of these systems. So it is important to say that we have different, for instance, this trajectory. This system was simulated 3 times, so we had 3 trajectories, 1 microsecond each. And to analyze the structure of polyamine, the conformation, I just remind that the structure of spermidine consists of 10 atoms in the backbone and also hydrogen atoms. The most important for the conformational dynamics is the dihedral angle, which we have here, which is formed by 4 atoms. And we have 7 dihedral angles. You can see they are numerated with Roman numbers. And there are 3 possible conformations for each dihedral angle. This is gauche plus, gauche minus, and trans. And as a result, we can have, in general, more than 2,000 conformations of spermidine for such small molecule. So we calculated the end-to-end distance. So this is the distance between these 2 atoms, N and N2. And we found that the distribution of these distances is not uniform. So we have 7 modes. You can see here these Gaussians. These Gaussians fit this curve. And we call these modes, conformational modes, by the analogy to the spectra, to the vibrational spectra. So, and we can determine the position of these modes, describing the distance between ends of polyamine. It is important to note that there are many theories describing the flexibility of chain molecules. So if we look at the textbook by Paul Flory, which is a well-known scientist, and he was the Nobel Prize winner, we can find that there is a well-known freely junction model with free rotation, hindered rotation model, and so on, different models. And also in this textbook, you can find some model which is developed for short polymers, because in all these cases, polymer is considered infinite, but also there are some models that describe short polymer chains. And if we take the parameters of spermidine, we can calculate that the end-to-end distance should be about 8.4 angstroms. And it is close to the distance, which is mean distance that we have for polyamines in solution without DNA. But when we add DNA molecule, you can find that we have different peaks, shoulders, and so on. We have additional modes, and we have at least 3 modes that appear in the presence of DNA double helix. So we have to understand the reason why they appear, and of course, this is it is very difficult to calculate such peaks, the appearance of such peaks with simple models that I showed before in the previous slide. So we obtained that we have some conformations of polyamines which are common for DNA system and for the solution system. You can see here the snapshots of these molecules that bind to DNA. They are localized in different regions and they bind to different phosphate groups in the minor group, in the major group. So we can see this directly from our simulations. And also there are band conformations, mode 1, mode 2, mode 3, where polyamine is essentially banned. And this conformation is stabilized by DNA. So it binds to DNA, and only in the presence of DNA molecule, this conformation appears. So we analyzed the most band conformation, mode 1. So you can see here, the distance is extremely short between ends of polyamine, and we analyzed the dihedral angles that we have in the case of this polyamine molecule. So you can see that in the ends and dihedrals, we have gauche always. So there is no trans conformations here. And also what is very important, we have always trans in the case of dihedral angle 5, it is here. It is here, or if you look at this scheme, you can see that dihedral angle contains this nitrogen atom. Other dihedrals can take different forms, and I would like to stress that we have 2 dihedral angles which have essentially distorted conformations. So you can see that there are different peaks, not gauche, not trans, and it means that this dihedral angle is not energy minima. Something makes essentially stressed this dihedral, and this is DNA. And to understand how it happens, how polyamines take such distorted conformation, we have to understand where polyamines are localized in the case of this distorted conformation. And we found, we measured the population of polyamines in the case of this conformation. We found that polyamines are localized mostly in some particular region, which is here. You can see in this diagram, this is thymine and guanine. So 16 and 31, you can see high bars of this histogram. And at these positions, the polyamines are almost in contact, in direct contact with oxygen atoms of phosphate group. You can see the distribution of a contact distance. It is 2.7. This is contact, very close contact. So, and if we analyze the structure of this system, we found that in this case, the oxygen atoms, different phosphate groups of different strengths, are very close to each other, and the polyamine hugs to these oxygen atoms. And you can see that this is in the case of trajectory 1, and the same we found in the case of trajectory 2. This is independent trajectory, and we found the same complex, so it repeats. And in this complex, we have very short distance between two phosphate groups, and this structure is stabilized by polyamine molecule. You can see the characteristic distances, they are very short. And of course, it will never happen if we do not have oxygen atoms, we do not have the phosphate groups. And the same phosphate groups will never take such conformation, very short distance, if the polyamine will be not present in the system. So we also analyzed the dynamics that can be in the case of this band conformation, and we can schematize this polyamine molecule with the phosphate groups as in such a way. You can see this interaction. Amino group 1 binds to oxygen atom. The middle oxygen atom amino group binds to the both oxygens, and the last one amino group binds to the oxygen atom. So we have two rings. One ring is 6 atom ring, and another one is 7. Taking this into account, we can use analogy with cyclohexane and cycloheptane, which are well known, and they are well studied. And we know that cyclohexane is stress-free cycle, and cycloheptane is always stressed. As a result, you can see here these atoms, and here we have stressed dihedral angles, and also these dihedral angles, which are always in gauche. It is in line with the conformations that can take cyclohexane and cycloheptane. So in the very end, I would conclude that the flexibility of polyamines is extremely important, and the conformations should be taken into consideration for the study of interaction of polyamines with DNA. And we found 7 conformational modes, 7 types of polyamine conformations that some of them appear only in the case of interaction with DNA. In particular, mode 1, which binds to DNA and essentially influence the local structure of the double helix that may have big importance for the DNA biological functioning. This research is published in Journal of Molecular Liquids this year, and also one paper is submitted to the Low Temperature Physics. Together with my colleagues, I acknowledge Professor Ato Laaksonen from Stockholm University, Francesca Mochi from Cagliari University, Leon Engelbrecht from Cagliari University, Dr. Tudor Vasiliu from Petru-Poni Institute of Macromolecular Chemistry in Romania, and Giuseppe Brancato from University of Pisa. I thank funds, computational facilities funds, in particular National Academy of Sciences of Ukraine, and thank you for your attention.
Thank you. Thank you very much.
My question is that in the biological cell, there are present also ions which interact strongly with DNA. What is your expectation how ions will affect binding of thymidine?
Thank you very much. This question is very interesting, and it is very complicated. It was studied previously in different papers. In particular, I cite these papers by Korolev, Lyubartsev, Laksonen. They studied this question 20 years ago, but at another level. So this is very short trajectory. So here, if we use 1 microsecond trajectory, in that times it was 2-4 nanoseconds. So this is another time scale, and it was extremely difficult to study this. But there is a competition between polyamines and small ions. Here you can see sodium. Of course, polyamines win this competition because they are big, they have a larger charge. But still, it is very important that small mobile ions are present here, and they influence the interaction with DNA. So, but this question is not completely studied. Yeah.
Okay. Thank you. Thank you very much.
Thank you. Thank you.